Hello dear students, today we'll be doing a very important topic of your CSIR-NET Life Science syllabus. The topic is inheritance of mitochondrial and chloroplast genes, and we will also be doing extra chromosomal inheritance, wherein we will talk about cytoplasmic or maternal inheritance. This topic most students leave out. Why? Either their concept is not clear, or they are afraid of the questions, or they think that they can skip some part, so they skip this part. But actually this part is not so tough, and the questions I will tell you at the end of the video what type of questions are important from this topic, and we will also discuss some questions. So stay till the end because there are many questions we will solve and we will do the topic very nicely today. So welcome back to Tamali's classes, and it's been a long time, and so today let's start with the class. So in today's class we'll be talking about the organelle genomes. We'll talk about the maternal inheritance and cytoplasmic inheritance. We'll be talking about the endosymbiotic theory and threshold effect. Also, we'll talk about the mitochondria and cytoplasm inheritance patterns, the mitochondrial DNA and the chloroplast DNA mutations, and also at the end we'll talk about some important topics for CSIR-NET exam and also some questions from this topic. Let's begin. So first we'll talk about the cytoplasmic inheritance. What is cytoplasmic inheritance? See, there are some traits which are transferred to the offspring from the parent through the cytoplasmic organelles. Okay, that means those organelles which are present in the cytoplasm. Usually we know that we get information from the chromosomal DNA, and that is inside the nucleus. So we have always learned about in sexual reproduction what happens, that the offspring gets the characteristics from the parent through the chromosomal DNA which is present in nucleus. But here we're talking about the organelles which are present in the cytoplasm, and from which there are some traits which are being transferred to the next generation, or you can say offspring. So here we're talking about cytoplasmic inheritance. So that means the traits are being transferred from the organelles present in the cytoplasm, not from the nucleus. Okay, so this is the cytoplasmic inheritance. Now this is maternal inheritance. See, maternal inheritance is also cytoplasmic inheritance only. What is the difference out here? The difference out here is that during sexual reproduction, what happens? We know that in sexual reproduction, normally the egg cell and the sperm cell, they fertilize, they form zygote, right? But here what is happening? In here, the egg cell which is containing the cytoplasm, okay, they will transmit, or they will send, okay, they will transfer the entire cytoplasm to the zygote. And the cytoplasm has got organelles. So along with the cytoplasm, all the organelles from the egg cell will be transferred to the zygote. But this sperm will not contribute any of the cytoplasm or organelles. It will contribute only the nuclear DNA, or you can say nucleus. Okay, only the nuclear DNA it will contribute. Therefore, that means what? That the cytoplasmic genes are passed down from the mother, only from the mother, not from the father. That is why this type of inheritance is known as maternal inheritance. Understood? So here there is no contribution of the sperm cell in giving the characteristics to the zygote, the cytoplasmic characteristics, okay, genes, cytoplasmic genes, but only they will give only the nuclear DNA. So this is the difference out here. Now we talk about the organelle genomes. As I have told that in the cytoplasm there are some organelles, and along with cytoplasmic genes, cytoplasm, some organelles are being transferred from the mother to the offspring. So here we'll talk about two organelles which are mitochondria and chloroplast. Why we're talking about these two organelles? Because these two organelles are high, they are, you can say that they are one of the most important, they're the most important organelles in the cell. All the organelles are important, but they play a very important role out here, and they are also very special. You know why? Because they have their own DNA. See, we know that nucleus has their DNA, but other than nucleus, there are more two organelles which have their own DNA, and that is mitochondria and chloroplast. They have their own DNA, they have their own ribosomes, and they have their own replication machinery also. Okay, so these two organelles, they are very special, and these are the organelles which we'll talk about today, their genomes and their inheritance pattern and everything. Also, what happens when there is mutation occurring out here. So if you see the diagram out here, you can see we can talk about the structure. So see, I already have a video dedicated to the cell and the cell organelles. So in this class, I will not talk about the cell organelles. This is mitochondria, this is chloroplast. Okay, so you can refer to my cell video where you will get the details about mitochondria and chloroplast. So here I will just tell you that mitochondria out here we're talking about the mitochondria in animal cell out here, and also mitochondria is present in the plant cell also. But chloroplast is exclusively present only in the plant cell, because chloroplast is used for photosynthesis, which animals cannot do. Okay, but mitochondria gives energy, which is required in both plant cell and animal cell. Now let's talk about mitochondrial genome at first. So these are some points that I've given out here. Okay, so these points are very, very important, and uh, objective questions can come from these points also. So this, see, I've given out your notes, written out notes out here for your taking, for your note taking. Okay, so you can take the notes from here also. That's why I always give written notes on my videos. Okay, so you can see out here, number one point, that mitochondrial genome is a purine rich. Okay, it have got two strands, or you can say it's a double stranded genome. Okay, so that means they have got two strands. So one strand out here is a purine rich heavy strand, and the other strand is out here a pyrimidine rich light strand. So since it's a double strand, so these two, both the double strands, both the strands are distinct. Okay, this is the nature. Now these mitochondrial genome, they have got 16,569 base pairs. Remember this. Okay, the gene capacity, they are 37 genes out here. So for when you talk about chloroplast genome, this will be different. Okay, now for gene distribution, so what are the gene distribution out here? We know there are 37 genes out here. So how it is distributed? So you see 13 proteins out here are for the respiratory chain, and 22 are tRNAs, and 2 are ribosomal RNAs. Okay, also mitochondrial genome, in this genome, they do not have any histones. So since they do not have any histones, what happen? They are more vulnerable to oxidative damage. Okay, when they have histones, they can protect themselves, but since they do not have histones, they're vulnerable to oxidative damage. Also the D loop out here, you can see the D loop out here. Okay, control region, it's also known as a control region. This D loop, it is a unique non-coding region. Okay, also known as a displacement loop. This acts as the main control site for replication and transcription. Also, in this mitochondrial genome, it's very highly efficient. Why it is highly efficient? Because they have got no introns. Okay, introns, you know what is the introns? Introns and exons, again, that I have taken a different class for it. So introns out here means non-coding sequences, okay, which do not code for any gene, any protein, okay, which could not code for any protein. So out here in this genome, there are no introns present. Okay, so see, you can see there are many genes. So these genes, between the genes, there are no introns. All right, that is why we say it's highly efficient. Talking about the functions of mitochondrial gene, what are the functions? Since we know about there are many genes out here which are coding for respiratory complexes. So there are 13 coding genes, okay, protein coding genes. So these 13 protein coding genes, you can see, they provide the blueprints for subunits of vital enzyme complexes, complex 1, 3, 4, and 5. So you know, these complexes are used in oxidative phosphorylation. All right, and for these complexes, these genes, they provide the blueprints. All right, then the RNA machinery, the 22 tRNA and the 2 rRNA genes, they allow the mitochondria to translate its. Okay, now we know that if they have got genes tRNA, RNA, we know that they are helping in the translation process, or they're helping in the protein formation. So since they have this gene, so they are also involved in translating its own proteins. They can produce their own proteins inside the matrix. Okay, now they are also having separate replication starting points for each strands. We know there are two strands, so heavy strand and light strand. So they have got their own origin of replication. So heavy strand will be known as origin of heavy strand replication, and light strand will be known as origin of light strand replication. Okay, these are the functions of mitochondrial genome. Now moving on to mitochondrial inheritance. Now we know that mitochondria, they generate ATP. Okay, what is ATP? ATP is also known as a currency, or you can say energy currency. Okay, that means it is providing energy to the entire cell. So now suppose if there is a defect in this mitochondrial DNA, you know, there are some parts of in a body which requires too much of energy. Okay, they're known as high energy organs, like our heart, our brain, our nerves, they require a lot of energy. So if there are any defects in mitochondrial DNA, then what will happen? It will have an impact that will affect these high energy organs. All right, now what is the mechanism out here? See, mitochondrial inheritance out here, what is the mechanism? So you know, during fertilization, what happens? The sperm, as I've already told before, the sperm will contribute only the nuclear head. Okay, we know that sperm contains mitochondria, okay, but they will contribute only the nuclear head. The egg cell which is present in the the egg cell which is having the cytoplasm, and the organelles, it will give the cytoplasm and the mitochondria for the zygote. We know the sperm cells, if you see the sperm diagram, you will know in sperm diagram there is a mitochondria present, but the sperm is not giving any mitochondria to the zygote. The egg cell is giving the mitochondria to the zygote. Okay. Now for pedigree rules, so this I will explain in my future slide. I'll just tell you one simple point out here. Okay, if a mother is affected, okay, if there is a mutation, if a mother is affected, it will pass it straight to all of his children. Okay, whether it's a boy or a girl, it will pass. But if a father is affected, it will not pass it straight to any of the children. Okay, it will not pass any character to any of his children. Okay, so this type of inheritance comes under the mitochondrial inheritance. If you see such a pedigree, and let's say if you see that type of structure, the diagram I'll show you in my future slides, you will know that it is a mitochondrial inheritance. Now, mitochondrial inheritance pattern. What is the pattern followed in mitochondrial inheritance? So first of all, it's a maternal transmission. Already told you that only the female, or you can say egg cell, will pass on the mitochondrial traits. Number two, no male transmission, as already told to you, affects both sexes. Okay, now if the mother has the condition, if the mother inherits the condition and the mother is affected, both the son and daughter will be affected. Already told you in my previous slide. The last one is highly variable expression. Okay, now in a family members there are many, in a family there are many members, and the severity of the disease and the symptoms might vary in all those members. Why? Because there's a phenomenon known as heteroplasmy, again, which I will discuss with you in my future slides. So because of heteroplasmy, these family members, they can have different symptoms and of the disease. Okay, and even different severity of the disease. Now, mutations in mitochondrial DNA. See, whenever there is a mutation in mitochondrial DNA, now there are some impact on the high energy requiring organs. Okay, so there are some conditions out here mentioned out here. So LHON is the labor hereditary optic neuropathy. The name itself, optic neuropathy. That means eye cells are involved out here. So it causes the degeneration of retinal ganglion cells. Okay, so there's also nerve involved out here. So it will lead to acute vision loss. So it will be passing on this. If the mother is affected, it will pass on to the child also. Okay, but if a male is affected, it will not pass on to the any children. Now next is LHIR. LHIR is Leber's hereditary optic neuropathy. Okay, so it's almost same if you see the first one and second one. I don't know, but they write it as LHON and LHIR. Okay, so anyways, it's same. It causes sudden vision loss in young adults. So MELAS, it's mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes. So this condition will affect the brain, the nervous system, and the muscles. Last is MERRF. It's myoclonic epilepsy with ragged red fibers. It causes muscle spasms, epilepsy, and ataxia. So these are some of the conditions which are occurring because of mutation in mitochondrial DNA. Moving on to chloroplast genome. Now this is again very important. Please understand it and please see it. You can take notes if you want to. I'm going very slow as possible. Okay. So chloroplast genome out here you can see the shape and size. So this is again single, or you can say circular and double-stranded. Okay. So single, circular, double-stranded DNA molecule. Okay. Same like the mitochondria. Okay. What is the size out here? 120 to 170 kilobases. You can see the size is larger than the mitochondria one. Okay. Then it has got a quadripartite structure. That means it has got 4 divisions. I'll show you how. See, you can see there are 4 divisions out here. Okay. See what? Sorry. I'm so sorry for this. This one. Okay. You can see here 1 division, here 1 division, here 1 division, and here 1 division. There are 4 divisions out here. Okay. So LSC, this is the large single copy. It is a long region and it contains some unique non-duplicated genes. And just on the opposite, we have got SSC, small single copy. It's the shorter region, but it also contains unique genes. Okay. Then we have got some inverted repeats, IR. So here it's IRA and IRB. So they are identical segments. Okay. But they are flanking on the sides of the SSC. Okay. So what they do, they are helping in stabilizing the genome. Okay. That is why they are facing in the opposite direction. Since they're opposite direction, they can stabilize the genome. Fine. Now we come to gene content. So now in here, what happened? There are many genes out here. Some genes are house genes for ribosomal RNAs, transfer RNAs, and protein subunits for thylakoid membrane and chloroplast ribosomes. You know, ribosco genes, even they are out here, okay? Like even they represent those genes also, okay? Now, copy number. Multiple copies of the genome are present in single chloroplast, allowing for high levels of gene expression. Since there are multiple copies, so the gene expression is also very high. All right. So this is about a chloroplast genome. I hope you have taken the notes. Next, we come to chloroplast inheritance. Now, we know chloroplast is present exclusively in the plant cell because only plant cells can make their own food and they need chlorophyll, and chlorophyll is produced in the chloroplast. So chloroplast carries out photosynthesis in plants and algae. Remember blue-green algae? All right. Now mechanism. What is the mechanism? So since we know that chloroplast and mitochondria both are coming under maternal inheritance, or you can say cytoplasmic inheritance, so they're again transmitted from the ovule, okay? In a, if you, I have a video dedicated to sexual reproduction in flowering plants. You can check that video of mine. You will see the structure of the plant and the reproductive parts, how they are shaped. You know, you will see the ovule structure also out there. So the ovule out here, the maternal part is known as the ovule, the female part, and the male is the pollen, okay? So out here, the chloroplast DNA is transmitted from the ovule part, not from the pollen part, okay? That's why it's coming under the maternal inheritance. Now, but there's an example for this type of inheritance, which you can see in 4 o'clock plant, also known as the Mirabilis jalapa. It's a very, very, it's a very famous experiment, or you can say it's a, that is a most spoken about experiment in this inheritance. So there's a scientist, Carl Covens, who discovered the leaf variegation, okay, in this Mirabilis jalapa. Okay, so I'll talk about this in my future slides. Okay, so just remember this, that Carl Correns, he discovered that leaf color, green, white, or variegated, it depends entirely on the phenotype of the maternal branch providing the ovule. That means the mother part. Okay, pollen from any branch color has no effect on the offspring's leaf color. That means whatever color the pollen is carrying, it doesn't matter. The color will always come from the ovule, the maternal branch. All right. Moving on to chloroplast inheritance pattern. Now, in this chloroplast inheritance pattern, so we know that chloroplasts, they are exhibiting non-Mendelian inheritance pattern. So it's also known as cytoplasmic or extranuclear inheritance. Why extranuclear? Because it is not in the nucleus. Okay, it's in the cytoplasm, so it's also known as cytoplasmic inheritance. Now, they are having this in this pattern. They are now mentioning, they have mentioned 3 inheritance patterns. Okay, number 1 is maternal inheritance, which is a uniparental. We do not need the paternal, we need only the maternal out here. We need only the egg cell, we do not need the sperm cell. Okay, so egg cell is larger than the sperm cell, or you can say pollen grain, and it will contribute most of the cytoplasmic zygote. Example already given, Mirabilis jalapa. Now there's another cytoplasmic inheritance pattern out here, which is known as the paternal. So paternal is the male one. Okay, so it's also uniparental. So when the paternal inheritance occurs, what happens? There is no role of the maternal cell, or you can say there is no role of the egg cell out here. So chloroplasts are inherited from the male parent exclusively. This occurs in gymnosperms, okay, like pine trees and conifers. So in this type of plants, paternal inheritance occurs. Okay. Now coming to biparental inheritance, where the chloroplasts are contributed by both the male and the female parts. The name itself says biparental. Bi means 2. Okay, so it results in the mixture of chloroplast in the offspring. This can also be seen in some strains of geranium and some algae. You heard of the name about Chlamydomonas. So this also shows a biparental inheritance of chloroplast. Okay. Now, mutations in chloroplast DNA. What happens if there's a mutation? So when there's a mutation, what happens? See, you can see basically like, you know, in nuclear, what happens in nuclear inheritance. In nuclear inheritance, what happens? There are, you know, SNP, you heard about SNP, there are that type of mutations. But in chloroplast, in the plant, in the cytoplasmic inheritance, what happens? The majority of the mutations, they are occurring because of the, they are the insertions and deletions, also known as indels. Okay, so when there's a DNA replication, if there's a slippage at repetitive AT rich sequences, it can lead to insertions and deletions, insert and delete of some sequences. Okay, so that is happening mostly in this type of inheritance. Okay, so now Carl Correns, he did an experiment in 1909. Okay, wherein he have used the plant Mirabilis jalapa, and he is a discoverer. You can say he discovered this leaf variegation. he saw that the leaves exhibit patches of green mixed with white or yellow if there's a mutation in the plant. This happens because you know what? Because we know that these plants, they have got what they do. They they have chloroplast, and chloroplast, they produce the chlorophyll. Now if there's a mutation, what will happen? The chloroplast production will be affected. It will disrupt that function of production of chlorophyll. Okay, so what will happen? This will lead to mutations, and if there's a mutation, we know that chlorophyll, because of chlorophyll, the leaves are green color, the plant is green color. Now if there is a mutation in the chlorophyll, then what will happen? The green color will not be, the plant will not be entirely green color. It will be mixed with other colors like white, yellow. Okay, so that's called leaf variegation, and this was discovered by Carl Correns. Okay, now other number two deficiency out here is your photosynthetic deficiency. As already told you, see that if the if the chloroplast is affected now, then and the chlorophyll is also affected, what will happen? If you have heard about the light reactions occurring in the plants, you will know that chlorophyll, they attract light photons. Okay, they attract photons from the sun. So now if they cannot attract those energy, if they cannot capture those, they not attract, they capture those photons. Okay, so if the chlorophyll is affected, chlorophyll A, chlorophyll B, they are responsible. There are many mechanisms, photosystem 1, photosystem 2. So that's a different part that comes in the photosynthesis part. Okay, so now basically what I'm trying to tell you is that if chlorophyll is affected, if there's a mutation there, then the chlorophyll will not be able to capture those light energy. They will not be able to capture those photons. Okay, if they do not do that, what will happen? It will result in the slow growth. Okay, it will result in the lethality of the plant. Okay, so it will affect the plant. Number three is cytoplasmic male sterility. So this is used in agriculture basically. What happens out here, especially in maize or rice breeding? So out here, this mitochondrial trait will cause pollen abortion, facilitating the creation of hybrid seeds. You have heard about production of hybrid seeds, right? So that is happening in the agriculture. And out here again, what is involved? The mutation in chloroplast DNA. All right. Now, this is again a very important part, protein sorting signals. Okay, so what are protein sorting signals? We know that mitochondria, chloroplast both can form protein. Okay, so these protein sorting signals, they are specific N-terminal amino acid sequences. Okay, there are some specific N-terminal amino acid sequences which will target the nuclear encoded proteins. That means there are some proteins which are encoded by like which are in like the nuclear encoded. Understand that? Okay, so those places are being targeted by these signals. Okay, and these signals, where are they? They are present in the mitochondria and chloroplast. So they will target those nuclear encoded proteins synthesized in the cytosol to the respective organelles. Okay, so once they deliver those, whatever they're delivering, once they deliver those proteins, then they will be removed from the places with the help of some proteases. Okay, so these sequences, what they do, these are signals, right? They are basically amino acid sequences. So once they are transporting what they're transporting, then they will be removed by the some proteases. So we will do this in details for mitochondria and for chloroplast. I hope you've understood this. Okay, sorting means what? They are sorting. Okay, they're trying to find, they are doing some work. Okay, so mitochondrial sorting. So mitochondrial sorting will occur when they have got a N-terminal amphipathic alpha helix, okay, rich in positively charged and basic and hydrophobic amino acids like arginine and lysine. So basically, this targeting peptide, okay, whatever peptide they're targeting, that is also known as the matrix targeting sequence or pre-sequence, okay, which is 15 to 50 amino acids long. So for mitochondrial sorting to occur, it will require a N-terminal amphipathic structures alpha helix, which is positively charged, and also it is having the basic amino acids known as arginine and lysine. That is okay. It should be a lot. There should be a lot of arginine and lysine. Okay. So now number two is translocation machinery. So what is happening out here? Okay. So these are recognized by the surface receptors on the TOM. Okay, TOM is again a translocase which is present in the outer mitochondrial membrane complex. Okay, and it is passing on to the TIM, translocase, on the inner mitochondrial membrane complex. Okay, so translocation machinery means from where to where it's going. Okay, so there are some surface receptors on the TOM. So from there it is identifying, and then from there it is going on to the TIM part, the TIM area. All right, coming to chloroplast sorting. So see, if you study about mitochondria na, in details, you will know about Tom and Tim. Okay. So chloroplast sorting. Now this will require a N-terminal sequence which is rich in serine and threonine, and it will have small hydrophobic residues, but it will be poor in glutamate and aspartate. It will form random coil structures. Okay, see, this random coil structures is not permanent; it's temporary, and it will be there till they interact with some organelle membrane receptors. Okay, now what is the targeting peptide out here? It is known as the transit peptides. The previous one was known as the matrix targeting sequence or pre-sequence. Out here, the targeting peptide is known as the transit peptide, and which is 20 to 100 amino acids long. Okay, now translocation machinery. What's the machinery out here doing? The transport is relying on the TOC, translocon on the outer chloroplast membrane, and TIC, translocon at the inner chloroplast membrane. Okay, so here from TOC to TIC. Now, endosymbiotic theory, very, very important. So this theory, you know what this theory is very interesting. It says that mitochondria and chloroplast, they were once prokaryotic microbes. You know why they say? They say that earlier what happened, you know, those big enlarged in eukaryotic microbes, they they engulfed the some prokaryotic microbes, but they did not digest it because those microbes were giving energy and they were helping in formation of food. Okay, so they did not digest it; they remained inside the cell. Okay, the eukaryotic cell, they it remained inside it, and then they form this mitochondria and chloroplast. Okay, so that is the theory which says that the mitochondria and chloroplast, they were once prokaryotic microbes, and there are some reasons why they say that, you know, because see, the mitochondria and chloroplast size, they are similar as the size of prokaryotes. Okay, and in both binary fission occurs, they both divide by binary fission, which is again the same technique used by prokaryotes, bacteria. Okay, now they have their own circular DNA, which is again a property of bacteria. They have their own ribosomes, which is the 70s ribosomes, and that 70s ribosomes have subunits 40s, 50s, and 30s. Okay, so now this is again similar to bacteria. They also have their own ribosomes. They also have the same subunits. Also, they have a double membrane structure. Now, from where it comes from? It is said that the inner membrane of the mitochondria and chloroplast, it resembles that of a prokaryote. What about the outer membrane? The outer membrane is coming from the host cell. Okay, when the host cell is engulfing the vesicles, they are forming the outer membrane in mitochondria and chloroplast. Though it is not so much, you know, reliable, but this is a theory actually, endosymbiotic theory. Fine. Now we'll come to heteroplasmy and homoplasmy. Remember we did this previously. Heteroplasmy, I spoke about the word. So now we'll talk about it. So heteroplasmy, again, this is a state in the mitochondrial DNA. Okay, so both heteroplasmy and homoplasmy, it's a state of mitochondrial DNA within a cell. So suppose, you know, the cell contains thousands, and there are many mitochondrial DNA copies. So when all of these copies are identical, it's called homoplasmy. Homo means same, hetero means different. Now when a cell is containing a mixture of different mitochondrial DNA variants, like healthy as well as mutated mitochondrial DNA, it is called as heteroplasmy. Okay, hetero means different. Fine. Now let's talk a bit details on it. So heteroplasmy, what happens? If a person is having this condition, he or she will have a mix of normal and mutated mitochondrial DNA. Okay, since it's a mixture. Number one. Suppose he has inherited this both normal and mutated, and he have got high percentage of mutated mitochondria, then the disease is more severe. Okay, it will occur, and this occurrence will be more severe. Okay, now because this proportion of mutated mitochondrial DNA is passed down by a mother. Okay, it's a random, this transfer of mutated mitochondrial DNA is random. Okay, during egg development, it's random. So when the siblings are formed, it can be boy girl, so they will have different clinical outcome, or they will have different severities. Okay, they might have the same condition, but their the severities will be different, the outcomes will be different in those siblings. Why? Because this transferring is randomly, it occurs. There is no that, you know, he will get 20, he will get 20. There is no sorted out. These things are not sorted out. So this passing on is random, so they will have different severities. Okay, this is called heteroplasmy. Threshold effect. Now threshold, we know the meaning of threshold. Okay, so similar meaning is out here, but the phenomenon is different. Threshold effect in plant genetics, in this part, it is a phenomenon where a specific trait or disease will manifest only when the proportion of mutated organelle DNA, that means it can be mitochondrial DNA, or it can be chloroplast DNA, when they will exceed a critical limit. See, everything when it is when it is below the critical limit, it's normal, but when it will exceed the critical limit, so that's called threshold effect. It's a phenomenon. Okay, only then when it exceeds that, only then the disease will happen. That is, we know only, right? That's called threshold level. So below, above it, disease occurs. Below it, it's normal. Now, very important, coming to pedigree analysis. I want you all to please focus on this slide, okay? Because that is very, very important. Okay, students get very scared when they see pedigree analysis, but it's not so scary. Trust me. Just look at it, okay? Now, we know that when we talk about pedigree analysis, this square refers to male and the circle refers to the female. So you see in outer, this number one stage, first stage, you can see the female is affected, but both the males are unaffected. So you see when the female is affected and they have got two siblings out here, male and female. You see the female is affected, both are affected. Okay, now see, they have formed, many siblings have been formed, and again see out here, the male is affected, no one is affected. Male is affected, no one is affected. Male is affected, no one is affected. So what do you understand by this? It's a mitochondrial inheritance, because we know that if a female is affected, all the siblings will be affected, but if a male is affected, none of the siblings will be affected. Okay, so this shows that it's a mitochondrial inheritance. So suppose in exam you get a pedigree chart, do not feel scared. Okay, read it properly, go through it properly. Okay, you see the result. What is the result outcome? You can, by seeing the outcome, you can know. And again you see here, mother is affected, all affected. Mother is affected. You see out here, mother is affected, but here not all is not affected, but most of them are affected. Okay, so this basically you understand that this one and you see this ones, okay, the male ones. Okay, now coming to mitochondrial versus chloroplast inheritance. This I've already done previously, but I'm just summarizing up for you. Okay, in a very short manner we will do that. If you look at the similarities, both are cytoplasmic, both follow cytoplasmic inheritance they both are cytoplasmic inheritance, and they are having non-Mendelian transmission. Okay, now see the organism, mitochondria, so all eukaryotics. Okay, chloroplast only in the plants and algae. Okay, genome size is smaller, larger size genome. Primary biological role, so it is involving in ATP synthesis, and here it's photosynthesis. What are the mutations out here? Since it's related to energy, so muscle weaknesses and metabolic issues, and if it will lead to energy depletion diseases. And suppose it's a chloroplast, it is what it will occur? You know, we have done CMS, then leaf variegation. Okay, this will be there also, affecting the capturing of light energy, remember? So all this will be the diseases or mutations or phenotypes. Next, nuclear versus extrachromosomal inheritance. So this is also just for your knowledge. So if the DNA is coming from the chromosome, it's nuclear inheritance. If the DNA is coming from the organelles in the cytoplasm, it's cytoplasmic or extrachromosomal inheritance. So location is chromosomes, they are the organelles. In what is the genetic contribution? So we know that in sexual reproduction, both mother and parents from both 23 chromosomes are coming, right? So 50% from mother, 50% from father. But in extrachromosomal, it's uniparental, either the paternal or the maternal. Okay, so out here, maternal is occurring mostly. Mendelian ratios, they follow Mendelian laws of segregation and independent assortment, but they are all non-Mendelian. Zygote cytoplasm, okay, the sperm, it is not, see, in the sexual reproduction, the sperm will contribute, okay, but it will not contribute any cytoplasm during fertilization. But in extrachromosomal, what will happen? The egg will or you can say it will supply the cytoplasm. It will provide the cytoplasm, okay, and the organelles. But in the nuclear inheritance, there will be no cytoplasm given by the sperm. Okay, sperm does not contribute to any cytoplasm. It will give what it will give. It will give the character. That we will do. That's already done in my other classes. So I will not discuss that out here. So example, what are the examples? So nuclear inheritance, Examples are eye color in humans, and also seed shape in Mendel pea plants. We have seen, remember, dihybrid cross. Okay, I already have a class dedicated for this. Please go through my plant genetics class. And for extrachromosomal inheritance out here, the mitochondrial disorders in humans and leaf variegation in plants. So now, what are the topics that are important for CSIR exam? Maternal inheritance, heteroplasmy, Mirabilis jalapa, there is experiment on this, endosymbiotic theory, genome size, pedigree analysis, and protein sorting signals. So there are many questions that come from these topics. So I have given out here 6 questions. Okay, there are 6 questions given out here. So I'm sorry, the numbering is a bit different out here. So this is question number 5. Okay, so sorry for this. Okay, so please forgive this mistake, but I hope you can see the questions out here. So the questions are given out. There are 6 questions out here. So I want you to solve the questions. Okay, so the first question, let's do a bit of some questions. So CMS already I've taught you in the class. So in CMS, the molecular determinants for CMS are family genome of each of the following organelles. Very easy. CMS, you know, I've already told you. In my slide, the answer is in the slide only. Okay. In pedigree analysis, again, this answer is there in my slide. I will not tell you because I want you to please read the question. But the answer is given in my slides. Okay. Even here, the third question, a plant breeder crosses a pale green branch female, okay, of Mirabilis jalapa with a dark green pollen donor male. So out here female and male is there, assuming that normal organellar inheritance. That means the cytoplasmic inheritance. What would be the expected leaf phenotype in the F1 progeny? F1 progeny means the first generation. So it's very easy again. If the female is having pale green, what will happen? The generation all will have pale green. Okay. So now question number 4. Question number 4 is which of the following genetic crosses and I'm so sorry. Which of the following genetic crosses and the results indicate strict cytoplasmic inheritance? So again, very easy. Cytoplasmic maternal means what? From the mother it will come. So whatever the mother will be having, the offspring will have the same thing, and they're saying strict. That means none of the other type of none of the other, like suppose after the first one, mutant female, wild type male. If it's a strict, then wild type should not be there at all. Only the female should be there. So I've already given you the hint of the answer. Okay, I will not tell you anymore. I want you to do it. Okay, and comment. You can put your answers in the comment box. Okay, I'll reply to you. The next question again out here, these are some very important questions which are, you know, C-part questions type. All right. So let me check one question. Okay. So the next question out here is okay, researchers time the chloroplast DNA reciprocal process have been occurring out here. So which one of them is the expected results? So again, when a variegated plant means what? Different variegated means different type of colors are there. Okay, green, white, and variegated, green, white, and yellow. Okay, so if it is in the female plant, what will be there in the progeny? So I will not tell you because the answer is, I told you the answer. So out here one of them is wrong and the other 3 is right. Okay, so you have to know which is right. So okay, B, when a green plant is used as a female plant, all progeny are green, obviously. So B has to be there in the answer. So if you see in the option, number 4 is eliminated. Only 1, 2, 3 is there. Okay, then now I will leave this to you. I will not tell you the answer. Very easy questions I've given here. Okay, now what is the structural or genetic feature that distinguishes a plant chloroplast genome from a human? Oh my god, very easy again question. Chloroplast genome difference from the mitochondrial genome. So what is the difference out here? What is the difference out here? If you see number 1, linear double and circular, it's a difference? No, because both are double stranded. Chloroplast gene contains IR and which are not present in, yes, because only chloroplast genome contains IR and mitochondrial genome does not contain IR. So that is the answer number 2. So I've given you some answers. I've not given you some answers, but I've given you hints. I want you all to solve the questions. Write your answers in the comment box and I will reply back to you with the correct answer if you are wrong. But I'm sure you'll do all the right answers. So if you like today's class, do share it with your friends. Click on the like icon and do subscribe to my channel for more videos related to your CSIR-NET exam. See you again in my next video. Thank you so much.